Hello and welcome to Biology 415. Today's lecture is going to be Chapter 1, Cells and Genomics from Molecular Biology of the Cell, 6th edition. The lecture is going to contain 20 questions that you will have to answer and return to me. And the questions are going to be distributed throughout the presentation in a written as well as in spoken form. So let's begin with the basic building blocks of DNA. So what are they? Basically, it includes the phosphate, the sugar, and a nitrogenous base that come together to form a nucleotide. The DNA strand is basically a polymer made of these nucleotides, in which the phosphate of the first nucleotide is covalently bound to the sugar of the adjacent nucleotide. So this forms a single strand of repeating nucleotides that each are distinct. Then, subsequently, this single strand can be used as a template to generate the complementary strand. As you can see on image C, the second strand is being formed, and in this picture the first four nucleotides are already built. C, T, A, G, which stand for cytosine, guanine, adenine, and thymine. As you can see, they're complementary. The A goes with T, C goes with G, and that is true in any normal DNA. So through a sequence of biochemical processes which involved a collection of proteins that zip through the template strand of DNA, they add the complementary nucleotides, therefore forming the double-stranded complementary strand of the original template DNA, which is depicted in image D. And then, of course, when it's packaged into the cell, the most optimal conformation of DNA is a helix, shown in image E. So now let's look at the a parental double-stranded DNA in the cell. Each strand in the DNA can be used as a template for a new strand. So again, a complex of proteins would separate the parental helix double strand into each complementary strand, and each complementary strand therefore will act as a template for the new strand to be built, just like it was shown on slide number 2. So the overall process is the parental DNA is pulled apart and each strand is being used as a template for a complementary strand to be made. As a result, we get 2 strands of DNA which are, in theory, if everything goes well, are perfectly identical. So overall, the bonds between base pairs are weak compared to the phosphate and sugar bonds. Therefore, each of the strands can be pulled apart without damaging the DNA, without breaking the backbone, and subsequently each strand can serve as a template for fresh DNA. Now, depending on the cell, depending on a signal for replication, the DNA replication occurs at different rates, and are controlled with different proteins, different transcription factors. Basically, these are collection of proteins that initiate DNA replication. But the universal basics of DNA include the simple truth that DNA is the information storage for heredity, and the template polymerization is the way the information is copied from cell to cell, from animal to animal. So we have established how DNA is replicated. But the information in the DNA has to be expressed. And the DNA information, the genetic information, is expressed through generation of proteins or a sequence of amino acids. So we have a process of replication, which is copying of DNA. Then we have a process of transcription, which is taking a strand of DNA and making a complementary RNA strand, which occurs in a cell nucleus, and then the transcription product of DNA, which is generally called messenger RNA, is transported outside of the cell nucleus, and it is translated into complementary protein sequence, amino acid sequence. So a single DNA can produce replicas or many copies of complementary RNA. Now, the RNA molecules are usually, in comparison to DNA, are short-lived, which ensures a tight control over DNA transcription and protein translation, and which overall is gene expression. So what is a messenger RNA? It is basically a single-stranded polymer molecule. Being single-stranded, it can be very flexible compared to DNA, and therefore it can form weak bonds with itself. So if there are complementary sequences within a single molecule, and a single molecule is flexible, for example, if we have a sequence of GGGG, it can make a bond with CCCC segment. So this allows the RNA to form very interesting and elaborate secondary structures. Therefore, these secondary structures can help the RNA to recognize and bind with other proteins in a cell. And by doing so, it can help catalyze certain chemical reactions when associated with appropriate proteins. Review question 1. What is a DNA backbone? What is it made of? And how is it put together in a DNA strand? Question number 2. Please list the constituents of a nucleotide. So here's an example of RNA produced in hepatitis delta virus. As you can see, there is a lot of complementarity within the same molecule, and it makes quite a few bonds between each individual nucleotides, producing a well-defined secondary structure. So this three-dimensional structure in figure B is represented with the backbone being the blue ribbon, and the bonds between nucleotides depicted as bars. So this particular RNA can catalyze RNA strand cleavage. So we've been talking about proteins, and obviously proteins are important and carry vital functions in a cell. But what are proteins? They're basically long unbranched polymer chains. They're formed by stringing together monomeric building blocks, which are called amino acids. These proteins carry information that is encoded in DNA, and again, each protein has a specific function. And altogether, besides water, proteins form most of the mass in the cell. So let's briefly talk about protein synthesis. This process is complex, and we will revisit it later on in the chapter. But the overview is as follows. The messenger RNA, which is the transcript product of DNA, is read in triplet nucleotides. Each triplet is known as a codon, and it codes for a single specific amino acid. Now, if we do a simple math, we have 4 nucleotides and 3 codons, 3 nucleotides per codon. We can have 64 possible codons, therefore 64 possible amino acids. But in reality, we have only 20 naturally occurring amino acids, which means there are certain triplet sequences that code for the same amino acid. So how is this code read? Well, it's read through another RNA, which is known as transfer RNA. And transfer RNA basically has an amino acid attached to one end and a triplet sequence to the other side of the molecule, which is known as anticodon. The anticodon recognizes the codon on messenger RNA and adds a specific amino acid to the sequence of peptide, which later turns into a functional protein. So this is basically done in a protein RNA complex known as ribosome. It is a very large protein RNA complex machinery that allows actual synthesis of the proteins in a cytosol of a cell. Review question 1. Please list 3 differences between RNA and DNA. So here is what it looks like. On the left, we have a general image of transfer RNA. As you can see, it has several loops. It has the anticodon on the bottom, and the linear structure is the messenger RNA with the codon. And on the top of the transfer RNA, as you can see, is a amino acid. Okay. On the right side, we have a general image of a ribosome. And as you can see, the messenger RNA is fed through the ribosome. The transfer RNAs come into a well-defined domains within the large subunit of ribosome, and through a catalytic reaction, the amino acids are bound together. And this process is repeated throughout the whole length of a messenger RNA, forming the whole protein encoded by the messenger RNA. So after the complete protein is synthesized, as shown in a previous slide, the protein is folded on itself, forming a 3D functional structure. Now, this functional protein can carry on its intrinsic functions. This particular protein, here the image A is depicting a ribbon structure of a lysozyme. A lysozyme is a catalytic protein that has an active site that cleaves polysaccharide chains. Okay, so this active site has a specific amino acid sequence that binds to the polysaccharide chain and catalyzes a covalent bond breakage in a polysaccharide molecule. Now we've been talking about a cell, and let's define the inside and outside of the cell. So basically, the cell is delineated by a cell membrane, which is composed of a very interesting molecule, which has a phosphate group, which is charged. We have a glycerol, just like in any other fats, and we have two chains of fatty acids. We have a saturated chain, which is the straight chain coming down from the red circle, and we have an unsaturated on the right side of that. Unsaturated basically means that somewhere between along the fatty acid we have a double bond between adjacent carbons. Now, we all know that oils or fatty acids do not mix with water, right? And polar molecules, such as salts or alcohols, they are mixed readily with water. So if we put a such molecule, which is known as phospholipid, it actually creates a very orderly layer in which the polar group, the hydrophilic head, which is composed of the phosphate and glycerol, tend to favor being in direct contact with water, and the tail, which is the hydrophobic group, favors to be away from water towards, in this particular case, in the image on the right, we have a layer of oil, we have the hydrophobic tails pointing towards oil, and we have a hydrophilic head pointing towards water. That creates a monolayer. Now, if we have a bilayer, as you can see, a spherical structure in the water, as you can see, the polar groups are always towards water, and the hydrophobic tails come together away from the water, therefore forming a bilayer. In this particular case, this bilayer is called a micelle, and this is the mechanism by which a soap, for example, works, and it carries oils from our hands when we wash our hands. Okay, so we have a phospholipid that encapsulates water insoluble molecules, and with water they're washed away. Now we've been talking about a cell, and let's define the inside and outside of the cell. So basically, the cell is delineated by a cell membrane, which is composed of a very interesting molecule, which has a phosphate group, which is charged. We have a glycerol, just like in any other fats, And we have two chains of fatty acids. We have a saturated chain, which is the straight chain coming down from the red circle, and we have an unsaturated on the right side of that. Unsaturated basically means that somewhere between along the fatty acid we have a double bond between adjacent carbons. Now we all know that oils or fatty acids do not mix with water, right? And polar molecules such as salts or alcohols, they are mixed readily with water. So if we put a such molecule, which is known as phospholipid, it actually creates a very orderly layer in which the polar group, the hydrophilic head, which is composed of the phosphate and glycerol, tend to favor being in direct contact with water, and the tail, which is the hydrophobic group, favors to be away from water towards, in this particular case, in the image on the right, we have a layer of oil, we have the hydrophobic tails pointing towards oil, and we have a hydrophilic head pointing towards water. That creates a monolayer. Now, if we have a bilayer, as you can see, a spherical structure in the water, as you can see, the polar groups are always towards water, and the hydrophobic tails come together away from the water, therefore forming a bilayer. In this particular case, this bilayer is called a micelle, and this is the mechanism by which a soap, for example, works, and it carries oils from our hands when we wash our hands. Okay, so we have a phospholipid that encapsulates water insoluble molecules, and with water they're washed away. So now that we've talked about basics of a cell, let's talk about what kind of shapes a cell can have. Now, in terms of simple cells, such as prokaryotes, that are much smaller than eukaryotes, which are our cells, we can have them in many different shapes and sizes, such as streptococcus. Basically small little circular cells. We have our well-known E. coli, which are elongated cells with flagella, and they can move very rapidly. We have tiny cells called mycoplasma, which are probably the smallest cells. And then we can have really long elongated spiral treponema cells. So these are basically the four different types of conformation of cells that we can have. And of course, some of them can be branched, but these are the basic forms. Review question 1. Where are polypeptides synthesized? Question 2. What's the difference between polypeptide and a functional protein? Question 3. What is a catalyst? So now let's look closely at what an E. coli would look like. E. coli is a simple cell. It has DNA, It has a plasma membrane, a cell wall, a structure that our cells lack, and ribosomes. Basically, it has everything that it needs to replicate. And of course, it has a flagella that it uses to move. So the picture on top is a cartoon representation of what an E. coli looks like. And on the bottom is an electron microscopy image of an actual E. coli. Here is an interesting bacteria which is known as Anabaeana cylindrica, which is a cyanobacteria, which means it photosynthesizes. In this image we have a sequence of cells. Now this is not a multicellular organism, but these cells can form multicellular filaments. Most of these cells are photosynthetic, but some of them become specialized for nitrogen fixation, labeled H, and some of them become spores, labeled as S. So now that we've talked about cells, let's talk about the major domains of living things. So in general, we can divide all living things into a group called bacteria, a group of archaea, and group of eukaryotes. Bacteria being our everyday E. coli. Archaea could be organisms such as sulfolobos, which are basically single-cell organisms found in the hydrothermic vents at the bottom of the ocean. And eukaryotes, well, us, yeast, and many other multicellular organisms. Well, of course, it's obvious that there are variations between species, right? Humans and E. coli. Well, obviously we're different. However, if you look at this diagram, and this is true for almost the entire genome, as you can see, there are many instances in which human DNA is aligned with E. coli DNA. So even though we are very different from the genetic perspective, we have a large percentage of overlap in our genes. Kind of scary, isn't it? So such similarity in genomic sequence between different organisms may be explained by the notion that genes are generated from pre-existing genes. And there are a few phenomena that are very well documented. One form of genetic variation is known as introgenic mutation, which is basically an existing gene can be randomly mutated, which in turn is a change in DNA sequence, through various types of errors, which occur mainly in a process of DNA replication. Another form of changes in DNA are known as DNA duplication, in which basically a pre-existing gene is accidentally duplicated. And that creates a pair of identical genes within a cell. And the interesting thing is that these genes from that point on can diverge, which means mutate and undergo evolution completely separately from one another, and through time result in completely different genes. Then we have DNA segment shifting, in which two or more existing genes can break off and rejoin, creating a hybrid gene. The fourth type is known as horizontal transfer, which is basically a transfer of DNA from genome of one cell or organism to another. Another way to transfer the genome from one organism to another is known as vertical transfer, but it's quite common. It is basically the genetic information being transferred from parent to progeny. Here is a cartoon representation of genetic innovation. The first one is intergenic mutations. So an error is introduced into the DNA. That error is not fixed, and in a progeny cell, that error becomes incorporated into the DNA. Now, most of these mutations are unfavorable and will result in cellular death, but some of them may be incorporated and down the line may result in a favorable outcome. The next one is gene duplication, and it's basically the same thing, the same gene being duplicated into two. And then, because they are now separate entities, they can evolve on their own and result in separate genes. The next one, the DNA segment shifting, as you can see, is fairly straightforward. It's a part of gene A is mixed with a part of gene B, and they result in two very different genes. And finally, the horizontal shift is, again, the gene denoted in red is basically transferred from organism A to organism B. And obviously, it starts coding for protein, which is intrinsic to organism A. So the gene duplication in the genome gives rise to a family of genes. And genes that are similar to each other in certain types of relationships are known as homologous genes. And the homologous genes could be either orthologs. Orthologous genes are basically genes that are slightly different but have been derived from same ancestral gene. And we have paralogs, which are paralogs genes are basically a set of genes that result from a duplication of a single gene and divergence of one of those duplicates into its own separate gene with its own separate functions. So here is a cartoon representation of orthologs and paralogs. As you can see, we have an ancestral gene on the left, gene, let's say, call it G. And we have speciation to give two separate species. So species A and species B, they receive a variation of this gene. They are basically the same gene, but with some degree of variation between each other. The paralogous, of course, is a duplication of a single gene, and through time, slight variations through certain mutations and certain other processes form two homologous genes that are slightly different with certain degree of similarities in function. Now let's talk about another way DNA can be transferred from one organism to another. Now, it is debatable whether a virus is an organism or not, because all it has is a set of proteins and DNA or RNA, nothing else. So in this case, on the left, we have a general image of bacteriophage. Yes, it looks scary, but the good news is that this virus infects only bacteria. So this virus is composed of a head, a collar, and a tail, and some adapter proteins that allow interaction of this particular virus with a bacteria. And all it does is it attaches to the bacteria and it injects its DNA into it. Now that DNA takes makes use of the intrinsic bacterial machinery to replicate the DNA, make proteins, and that DNA, of course, is integrated into the genome of the host bacteria. And once this DNA is replicated and transcribed and translated into proteins, the DNA is packaged into its appropriate proteins that code for the head, collar, and the tail, and the virus is later released from the infected bacteria as a fully functional virus. So here is the image of a genetic material of E. coli. That circular structure is its DNA, okay? And just like many other prokaryotes, the genomic material in this cell are circular and make a loop. The yellow and orange basically depict the genes that encode for proteins, and the green arrows are regions that encode for RNA. In this circular genome, some genes are encoded from left to right, clockwise, and counterclockwise. And that is depicted with arrows on the circular DNA. And that is very unique for bacteria. And this is actually a very good tool that scientists use to do genetic modifications of cells and do genome research. So this is a fantastic, very, very, very powerful genomic tool, because once this circular DNA is inserted into another cell, such as a eukaryotic cell, that cell will start synthesizing proteins that are encoded in this circular DNA, which means we can insert DNA of interest for our own protein of interest, and the cell will begin expressing that protein, and we can conduct our set of experiments on that particular protein on those particular cells. So let's compare and contrast a eukaryotic cell now. The DNA in a eukaryotic cell is packaged into in a nucleus, which is surrounded by nuclear envelope. That is a double-stranded membrane, and it separates the DNA from the cytoplasm. The eukaryotic cell has what's known as cytoskeleton, which is a combination of proteins that provide the mechanical strength to the cell These proteins crisscross the cell from all over the points, and they provide the, as I said, mechanical strength. They control the shape and locomotion of the cell. The eukaryotic cell can be 10 times larger in linear dimensions compared to prokaryotes, and they can be 1,000 times larger in volume. So, just to put it in perspective. Some eukaryotic cells could be quite vicious. Some of them can be very, very effective predators. For example, neutrophils are a part of our white blood cells, part of our immune system. They can engulf other cells. In this particular case, neutrophil is engulfing or phagocytosing a red blood cell, which has been treated with antibodies, basically marked for destruction. Here is another eukaryote, which is a single-celled organism that eats other cells. Didinium. It's obviously carnivorous protozoan. It belongs to a group known as ciliates. So one of the more modern and more or less accepted theories of how the eukaryotes came to being is basically symbiosis. The predatory nature of eukaryotic cells helps explain why these cells are larger and have many different organelles. For example, mitochondria. It's an interesting organelle. It's basically have small bodies in a cytoplasm. It is enclosed by a double layer membrane. It can take up oxygen and basically generate ATP, adenosine triphosphate, which is the basic unit of energy for the cell. So it takes up oxygen, takes up sugars, oxidizes sugars to harvest energy, and then uses that energy to create ATP. The mitochondria is actually about the same size as a small bacteria. It has its own genome, and it is in the form of circular DNA. It has its own ribosomes that are very different. Well, they are different from other ribosomes in a eukaryotic cell, and they do have their own transfer RNA. So it seems as though it is an individual organism or individual cell living inside a larger eukaryotic cell. Quite peculiar, isn't it? So here is an electron microscopy image of a mitochondria, and on the right we have a cartoon depiction of mitochondria. The important feature of mitochondria is this branched and intricate membrane that penetrates into the mitochondria. We will talk about why mitochondria has this kind of a membrane structure later on in the course, but in general, this structure is very important at generating energy for the cell. So here is a short tale of mitochondria. Once upon a time, a mitochondria originated from free-living oxygen metabolizing, otherwise known as aerobic, bacteria that were engulfed, phagocytosed, by an ancestral cell that could otherwise make no such use of oxygen, or otherwise it was anaerobic. Now, mitochondria somehow escaped digestion, and it created a symbiotic relationship with the engulfing cell and subsequently its progeny. Now, mitochondria received shelter and nourishment, and in return provided power for the host cell. So this relationship between the primary anaerobic predator cell and an aerobic bacterial cell is thought to be established about 1.5 billion years ago. That's when the Earth's atmosphere first became rich in oxygen. So here is the tale of mitochondria in a cartoon representation. So we have the anaerobic cell that basically has a nucleus and basically nothing else. It phagocytoses a bacteria that is able to make use of oxygen in the environment. And as this bacteria is phagocytosed, it takes a portion of the plasma membrane of the phagocytosing cell. And then through a sequence of events, the mitochondria becomes a symbiont. It becomes a part of the cell. It is passed on to the progeny of the phagocytosing cell as a fully functional organelle. Now, another interesting structure that is similar to mitochondria is chloroplast in plant cells. Chloroplast has basically chlorophyll-containing membranes which can absorb solar radiation and make sugars for the plant to basically metabolize. And the idea here is very similar to that of mitochondria. So the underlying idea here is that some cells decided that, well, mitochondria is not enough. There are other sources of energy. And they engulfed photosynthetic bacteria, which started again absorbing solar radiation, started using the CO2 and water from within the cell, and started generating basically sugars for the cell to live on. Those sugars later are being uptaken by the mitochondria, and mitochondria generates the adenosine triphosphate, which is the energy molecule for the cell, at least in this model of photosynthetic cell. So in the modern eukaryotes, there are many genes from mitochondria and chloroplast that have been lost. But in reality, they haven't really been lost, but they have been incorporated into the genomic DNA of the host cell. The nuclear DNA in human cells contain a lot of genes that are essential for mitochondria. In plants, same thing applies to genes for chloroplasts. In both cases, the DNA sequences of these nuclear genes show evidence of their origins from bacterial ancestors. Well, maybe it is true. So the modern molecular genetics has been proven to be a very powerful area of study and a very powerful tool. And there are several species that are responsible for it. The model species include C. elegans, which are basically worms, fruit flies, Drosophila, zebrafish, mouse, and of course human cells. And these 5 animal groups altogether have laid bases for modern molecular genetics. Review question 1. Both the mitochondria and chloroplasts have lost some of their essential DNA. What happened to the essential DNA in these organelles? Well, now let's talk about each one separately. The C. elegans is the first multicellular organism that has had its complete genome sequenced. It's a small nematode, about 1 millimeter long. It lives in soil, and the individuals can be hermaphrodites that produce both eggs and sperm, which makes the generation of the progeny quicker and easier. Therefore, we can study the gene mutations and passage from parents to progeny much easier at much shorter time frame. Now, our Drosophila. The genetic studies done on Drosophila have revealed the fundamental mechanisms of animal development from fertilized eggs into an adult. Using Drosophila genome, we have determined that when we select for a particular gene, we inadvertently select for other traits also. And we have discovered that there are some genes that are simply linked. They are too close to each other and cannot be separated, which is one of the main mechanisms that purebred dogs that are bred for a particular gene often develop certain disorders. This is because when we select for a particular gene, another gene is selected with it. Zebrafish is the model for vertebrate development. These are convenient because, first of all, they're transparent, and we can easily see cell movement through the development of the embryo, and the embryo starts developing 24 hours after fertilization. So it is very convenient in terms of time to study a whole course of embryonic development. So the mouse and humans, surprisingly, are very similar in genetic composition. And again, with mice, the gestation period is about 28 days, which means we can have a progeny in less than a month. And we have genes and we have gene mutations that are actually the same between mice and humans. For example, in these pictures, we have this white patch on the forehead on both mice and this little baby, and both are a result of a mutation in the same gene, the gene called KIT. This gene is responsible for development and maintenance of the pigment cells. and mutations thereof results in lack of pigment in, well, the skin or the fur. This mutation is not really that bad, but it delineates the astonishing similarities between mice and humans. So to put the genetic similarities in perspective, we have this little graph here in which we compare the percentage of amino acid identity in a hemoglobin alpha chain. Animals and chimps. We know that chimps are very, very similar to us, but did we know that the hemoglobin alpha chain has 100% similarity between humans and chimps? Now, for example, humans and lamprey. I mean, come on, lamprey. It's an animal from Proterozoic era. I mean, millions, hundreds of millions of years ago. And yet, our hemoglobin alpha chain is 35% identical to that of lamprey. Scary. So, in summary, the eukaryotic cells have DNA which is separated into its own organelle, the nucleus. Eukaryotic cells have cytoskeleton, which are polymers of proteins that provide cell structure, motility of the cell, and they define the shape of the cell. Eukaryotic cells are complex and they have elaborate intercellular components. As we saw, they can engulf other cells, metabolize them. So eukaryotes may have originated as predatory cells that fed on other cells. And it is a simple truth that mitochondria and chloroplasts have their own DNA, and they may have been taken up by these predatory cells sometimes in the past, after which the mitochondria and chloroplast were able to avoid destruction and remained in the cell as symbionts. So all in all, eukaryotes are up to 30 times larger in their genome than prokaryotes, and they have thousand times more of non-coding DNA. Now, non-coding DNA doesn't mean junk. Non-coding DNA has a very important role in gene regulation. These regulations are essential for complex multicellular organisms. But many eukaryotes are unicellular, and some of them have been used as models for genetic studies. Other organisms that have been used and are being used as genetic models are a worm, a fly, a fish, a mouse, and also a milkweed as a plant model. Thank you very much for watching this webinar. I hope you enjoyed and paid attention. There were at least 10 review questions asked throughout the presentation. Some of them are in a written form and some of them in a spoken form. Please answer these questions and subsequent questions in the lessons throughout this week and return them in to me via email by Friday, by the end of the day, with, again, the name of the course, the name of the lesson. In this particular case, it would be the cell part 1, chapter 1, and your name. Again, thank you very much for your attention. Have a good week.